Hello, everyone, and welcome to today's webinar, Candida auris, Strategies for Preventing an Outbreak. I'm Susie Valdez of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by DiaSorin Molecular. To learn more about our sponsor, please visit them at molecular.diasorin.com. So let's get started. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. All the questions will be addressed by the speaker via the contact information you provided at the time of registration after the presentation is over. If you have any trouble seeing or hearing this presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Omai Garner, an Associate Clinical Professor and Section Chief in the Department of Pathology and Laboratory Medicine at UCLA Health System. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Welcome, Dr. Garner. You may now begin your presentation.
Thank you, Susie. So I'd like to begin by thanking Labroots and DiaSorin for hosting this seminar. I think that this is a very important topic, not only to clinical laboratories, but also to hospitals, infection prevention areas, nursing homes, really anywhere where you could find this particular pathogen. So as Susie mentioned, the talk today is called Candida auris, Strategies for Preventing an Outbreak. So to begin with some disclosures, I have research funding in my laboratory from relevantly bioMérieux, DiaSorin Molecular, and Beckman Coulter, and then I have taken honorarium for speaking from DiaSorin Molecular. So I really want to begin with this superbug. So as you can see from these headlines, we've really had a lot of press around Candida auris. And just to read these, so from Health, a deadly superbug fungus called Candida auris has been detected in 12 states. Here's what you need to know. This drug-resistant fungus is spreading. Scientists warn of new superbugs to come. And this superbug is a serious global health threat. Here's what you need to know about Candida auris. I think that this is unusual. This is not how we have handled overall outbreaks in the past. And I really think that it speaks to not only the seriousness of this infection, but also our global understanding of outbreaks and how we can come together, even as a public, to fight these types of diseases. So we have three objectives for the talk today. After the talk today, I would hope that the audience would be able to discuss the spread of Candida auris through a healthcare system, describe how microbiology laboratories can assist in limiting Candida auris infections. And this is going to be a big part of this talk. I really want the takeaway to be that folks working in a clinical microbiology laboratory are part of fighting this particular outbreak. And then the audience should be able to detail the challenges in accurately identifying Candida auris from culture within the laboratory space. So as an outline of the talk today, we're going to start with a clinical case that really highlights the deadly nature of this superbug, and then move on to a hospital-associated case. So figuring out an understanding on how this bug moves around a hospital and really can put patients in danger. Then we'll move on to detailing the global spread of this organism since 2009. Then I'm going to move into the microbiology and really talk about an introduction to the organism, talk about how we identify the organism, what it looks like on the plate, what susceptibility testing results look like for Candida auris, and then what the microbiology laboratory can do to help stem this global outbreak. And this is going to include concepts of passive surveillance, or things that's going to be done in the laboratory, or active surveillance, which is really key around how you can appropriately screen patients who are colonized for this organism. So let's start with the clinical case. So this case is published. The reference is on the bottom of the screen. A 63-year-old man presents to a Malaysian hospital with abdominal pain, weight loss, and fever. His complete blood cell count shows pancytopenia, including leukopenia and neutropenia. He started on IV antibiotics, and he has blood cultures drawn, all of this normal through a potential sepsis workup. Day 1 and day 4 blood cultures are negative, but the day 9 blood culture is positive by gram stain from the bottle and reveals yeast-like cells. So this is really the first clue for the clinicians involved in this case that potentially there's a yeast causing this disease. And so at that point, they start IV fluconazole. IV fluconazole is a normal treatment for yeast identification in the blood because typically the number one yeast causing candidemia is Candida albicans. And Candida albicans can be seen as predictably susceptible to fluconazole. So in this case, they're guessing it's potentially Candida albicans and putting fluconazole on board. Unfortunately, despite antifungal treatment, the clinical condition deteriorated. By day 15, because the patient was getting worse, they added meropenem. Meropenem is added to be able to cover potential unusual gram-negatives or resistant gram-negatives that could be causing infection. By day 16, the patient showed a brain bleed, but surgical intervention wasn't possible due to the low platelets, so they couldn't relieve that pressure or that bleeding. And then by day 18, unfortunately, the patient had passed away. So I want to dive into the microbiological workup of this particular case because an organism identification didn't come about through this clinical case that could have led to different treatment. So looking at the microbiology workup, on day 9, you'll remember the blood culture bottle was yeast positive on the gram stain. They subcultured that blood culture bottle to sabro-dextrose plate. This is a plate specifically to grow yeast or fungus. And after 48 hours, a yeast-like colony grew. Now, at this point, because the laboratory itself wasn't able to do full yeast identification and fungal workup, they sent the plate out to a reference laboratory for further organism identification. And I think that this is common practice in a lot of microbiology laboratories. The ability to do yeast identification on hand isn't something that all labs can do, but most labs have some kind of send-out lab they can move the isolate to for identification. Unfortunately, when that happens, though, you see delays in overall organism identification. So at this point at the reference laboratory, 48 hours later, a cream-colored smooth yeast colony was growing on this sabdextrose plate, and it only appeared to be one morphotype. So it appeared there was only one type of yeast there. This was also plated onto chromogenic agar. The chrome agar specifically can identify different species of Candida by the color that they show up with. And what was interesting in this case is that even though on the SDA it appeared there was only one type of Candida there, on the chrome agar there were two different colors. And so you can see this represented by picture. So in A, there was one isolate showing up greenish on the chrome agar. They labeled UZ1446. And then there was isolate B showing up pink or pinkish mauve on the chrome agar, isolate UZ1447. And they held that second isolate to 120 hours, and you could see it develop this purple color on the chrome agar. So this was really necessitating a workup of two different yeasts that were found in the blood. So one of the initial workups is just to look at the colonies specifically under the microscope. So they isolated yeast cells, and what they saw in either colony was small budding yeasts. You can see this on A and B. And then there are multiple identification schemes to be able to figure out what type of yeast it is and to identify that type of yeast to species. One of the things that you can do is you can grow the organism on cornmeal agar and look for pseudohyphal formation. Specific types of pseudohyphal formation can lead to a potential identification of the organism. So isolate UZ1446 on cornmeal after 48 hours showed long branched pseudohyphae. but isolate UZ1447 after 48 hours on cornmeal, no pseudohyphae was observed, being able to put these yeast in categories for identification. They also proceeded with identification systems within that laboratory to identify. So some of these were biochemical or carbohydrate based. So looking at the first isolate, this UZ1446, you can see that the API 20C called it Candida tropicalis at 99%. The Vitek 2, which is another yeast identification, also called it Candida tropicalis at 99%. This fit in well with what was seen on the cornmeal. Candida tropicalis is known to produce those long pseudohyphae. All of this matched for an ID. The other organism was a bit confusing, though. So the other organism, remember, no pseudohyphae. The API 20C called it 99% Rhodotorula glutinus. Now, this ends up being a little bit of a problem. So I have a picture on the bottom of what Rhodotorula looks like on the plate. It's a red-colored yeast. And if you'll remember, the yeast that was identified, UZ1447, I give an example of that plate below. It's a cream-colored yeast. So right away, this is leading to questions in the potential identification. Another system was run, the Vitek 2, and it called it Candida hemolonii. So Candida hemolonii matches a little bit better with what was seen from the picture on the yeast, but now you have a distinct discrepancy between two identification systems. And so later in the process, so all of that took about 4 days, as you can see on the right side, but we still don't have a good identification. So they moved to sequencing. And so in moving to sequencing, what they found is that in ITS sequencing, it confirmed the Candida tropicalis, but what was revealed about UZ1447 was that this isolate was actually a Candida auris, and this was also confirmed by a different sequencing method. Moving on to susceptibility testing, what was found that when you did susceptibility testing of the Candida tropicalis, what you saw is that whether it's amphotericin, taspofungin, anindulafungin, any of the echinocandins, or all of the azoles, fluconazole, itraconazole, voriconazole, they all tested as susceptible. So the fluconazole treatment that the patient was put on, when the gram stain revealed yeast in the blood, would have covered this particular organism. The problem is that when that testing was performed on Candida auris, and later on we'll get into the talk about susceptibility testing for Candida auris, what was found is that the Candida auris was resistant to all of the antifungals, but also especially resistant to the antifungal that the patient was placed upon, which was fluconazole. So this ended up being a fatal case of Candida auris in mixed candidemia with Candida tropicalis in a neutropenic patient, just showing how deadly this bug is and how resistant it can be to antifungals that are put on board. So I want to take a step back then and talk about the global emergence of this infection. Candida auris was first isolated from ear discharge in 2009 in Japan, and at that time it was a very difficult organism to identify. Then a study from South Korea identified 15 patients with Candida auris otitis media in 2009. And you can see either ear infections or ear discharge. This is where this Candida gets its name. It's why it's called Candida auris. Interesting, though, in that same year, there were nosocomial or hospital-associated infections reported in South Korea. Then, over time, the organism has continued to spread around the globe from 2012 to 2019. And so I have a picture, a global map in Current Opinion of Microbiology, published in 2019, showing that in 2009, you really only saw Candida auris infection in Japan, in South Korea, but at over time, 2013, 2016, 2018, we saw this distinct spread of this organism globally. So how is that happening? You know, how is an organism spreading so dramatically around the globe? And I think that highlighting how it spreads through a hospital system can show how it spread as infected patients move their way around the world. So we're going to use an example from the UK. So there was an initial case of Candida auris that was reported at the Royal Brompton Hospital in London, and this is a 20-bed adult ICU facility. The following week, the patient in the adjacent bed in that room was found to be Candida auris positive from a sputum specimen and developed a line infection. Subsequently, then, 9 other cases were identified, and when environmental sampling was done, it showed contamination with Candida auris all around the room. There were similar patterns of introduction and local spread of the organism that have reported in the U.S. and around the world. So you can really see that healthcare centers end up being areas where an infected patient can spread this organism not only to other patients causing infection, but also through the environment. So this brings up the question of infection and colonization with this organism. I really want to start with clinical infection. So what typical disease types do we see reported for Candida auris infection? And this is from a fantastic clinical microbiology reviews called Candida auris, a review of the literature in 2017. And what was seen in this table is that there were 291 reported cases of candidemia by Candida auris, or Candida found in the blood. And this has been the most common site of infection for Candida auris. But it's also been associated with other types of infections. So it's been isolated from central venous catheter tips, CNS, ear, nose, and throat, respiratory tract, urogenital system, abdominal infections, skin and soft tissue infections, including surgical wounds, and even in the bone. So this organism doesn't just exclusively cause disease in the blood, but can also cause disseminating and devastating disease in other areas of the body. So then what type of patients are associated with Candida auris infections? So they're found most often with invasive Candida auris infections in patients who are critically ill, patients who are found in intensive care, patients undergoing invasive procedures, patients with serious underlying medical conditions, patients with hematological malignancies, immune suppression, broad-spectrum antibiotics, patients with medical devices like catheters, CVCs, or urethral catheters, and patients in long-term care facilities, including nursing homes. So what's interesting about this is that this is also the risk factor group for any invasive Candida infections. And yet the mortality rates from Candida auris infections that are invasive can be up to 50%. So higher than we've seen potentially in other Candida infections, and a lot of that has to do with the extended resistance. We can contrast infection with colonization. And so there's been a link between patients with invasive infection being colonized. So what area are these patients colonized with this organism? So Candida auris has been grown from patients with invasive infection from the nares, from the groin, from the axilla, and the rectum. It has also been isolated for up to 3 months or more after the initial infection. So what's interesting there is that even if the infection is cleared, the Candida auris can still be colonizing that patient. As we saw in the hospital infection in the UK, it can also be isolated from the environment of colonized patients, and it appears patients who colonized with Candida auris actually successfully shed this organism into the environment. So then it's interesting to think about contact time. How long would it take for a patient to shed Candida auris from the environment, and you test for it in the environment? So contact time for acquisition of Candida auris from a colonized patient or from the environment has been shown in as little as 4 hours. So a patient moving into a room that has environmental contamination can be colonized with that organism very quickly. And then invasive infections after that colonization have been acquired from contaminated areas within as little as 48 hours of an admission. So a very aggressive organism in not only its ability to contaminate the environment, but also to colonize a new patient and cause invasive infection in that patient. So again, this all then contributes to an unsurprising spread and global spread of Candida auris. You know, we live in a very connected world. You know, I think we see this with a lot of outbreak organisms. We've seen this in our recent ongoing Ebola outbreaks, that people move around the world and bring infections with them. And so it's not a surprise then with an organism that is so ubiquitous in either its ability to colonize or actually live in the environment that you can see spread of this organism globally. So I wanted to look then specifically at some of these outbreaks and how Candida auris can become the predominant invasive infection in an outbreak location. So let's look first specifically at a Spain outbreak of Candida auris in 2016. And so invasive Candida infections were looked at pre the outbreak and post the outbreak. So before the outbreak, what you saw is that most of the invasive infections were associated with Candida albicans at 31%, Candida parapsilosis at 46%, and again, these organisms can usually be treated with fluconazole. And then, of course, there was Candida glabrata and Candida tropicalis representing a lower percentage. But then after the outbreak, and this is, I think, where this number becomes shocking, 42% of the invasive Candida infections were associated with Candida auris. So now, again, a population and an organism that can't just be readily treated with fluconazole. So it's almost like Candida taking over for the invasive infections after an outbreak, which makes it so important for an area to stop an outbreak from happening in the first place. We can also, unfortunately, see similar numbers from outbreaks of Candida auris in South Africa. So here on the y-axis, we're looking at the number of overall cases of candidemia in South Africa in 2016 and 2017. The number is 2988. These are private sector hospitals, and you can see that during their outbreak setting, even though parapsilosis was number one, Candida auris was the number 2 cause of candidemia in South Africa, making mortality rates go up, making treatment very, very difficult. So I want to then look specifically at the global spread of Candida auris, looking at the CDC website. And I would just encourage everyone listening to this, the CDC actually has a wealth of information on Candida auris. If you have any other questions after this talk, I encourage you to go to the website, and I've tried to highlight this website as we go through to learn more about Candida auris. So this is a map showing the countries from which Candida auris cases have been reported as of August 31st, 2019. And so what you can see, even compared to the other maps that I've shown, is that this organism continues to spread. So first, looking at the light green colored cases, these are countries where there's a single Candida auris case that's been reported. So you see this in Chile, you can see this in Turkey, you can see this in parts of Southeast Asia. And what we've seen before is that these countries that have one case quickly progress to countries that have transmission or multiple cases. So the countries in light blue are the countries that have had transmission or multiple cases of Candida auris reported. You can see in North America, this includes the United States and Canada, South America, Colombia, Venezuela, South Africa, many countries in Europe, many countries in Asia. In addition, because this is a CDC map, it also shows countries where U.S. Candida auris cases are linked to healthcare stays. And so these are patients that had healthcare stays in different countries, came back to the United States, brought Candida auris back, and continued outbreak infections in these areas. And you can see that includes Colombia, South Africa, India. Many countries have contributed. So it just kind of shows how this global spread is continuing. I want to take a deeper dive then into infections in the United States. And so this is a U.S. map of clinical cases of Candida auris reported by U.S. states as of March 31st, 2019. And so you can see there have been 3 states that have reported 100 or more Candida auris clinical cases. This includes New York, New Jersey, and Chicago. There's a state that has between 11 to 50 infections. This includes Florida. And then moving on down, California, Texas, Oklahoma have seen between 2 and 10 clinical cases. And then other states like Indiana and other states have shown other lower levels of infection. I think what's important to realize here is that an infectious case vastly underestimates the number of colonized patients. And what we've seen in this outbreak is that if you have someone with Candida auris in their blood and you start to do patient surveillance around that person, room surveillance, and hospital surveillance, you find many times that number of patients that have already been colonized. So looking specifically in detail at some of the spread in the United States, there have been 725 confirmed and 30 probable cases of Candida auris as of June 30th, 2019, but twice that number. So 1,400 total patients have been found to be colonized. Those colonized patients, of course, are now highly at risk for invasive Candida auris disease. Most of these cases have been associated with either hospitals or long-term care facilities like nursing homes in New York City, New Jersey, and Chicago. But as I mentioned, 9 other states have reported outbreaks. Looking into the details of one of the New York outbreaks in 2017, it included 166 clinical cases, 184 colonized patients, and they all came from one source patient, one source identification of Candida auris for that entire outbreak. So you can see that this represents and I won't even say potential, this represents a public health emergency. And why this could expand is if we look at that table over the number of patients we have in our states that are in registered nursing homes. And so this is from 2017 data. It estimates overall there's 600,000 people in registered nursing homes, and we have 100,000 just in California. This represents a population that, if Candida auris can take a hold, cause an outbreak, and colonize in that area, all of these patients could potentially be colonized or infected. So I hope that the last few slides have sort of highlighted the immediacy of the issue and why we really need to look into what we can do to stop the further spread of Candida auris. So now I want to spend the next few slides introducing you to the microbiology of this very interesting organism. So Candida auris, as we saw in the clinical case, grows as a nondescript white to cream colored colony on SDA. These pictures are pictures that came from the CDC website, and so that's the top picture. And the bottom picture, pink to beige colored colony on chromogenic agar or chrome agar. What's interesting about the nondescript white to cream colored colony is that that is what most Candidas look like when they grow up on a plate. So its initial growth is not a clue to whether or not it's Candida auris. It's ovoid to elongated budding yeast cells on cornmeal agar without the formation of pseudohyphae in most cases, and it grows well at both 37 and 42 degrees. It's interesting to look at the genome. So it has a haploid 12.5 megabase genome with between 6,500 and 8,500 protein coding sequences. And when an analysis of those sequences is done, there are many genes associated with virulence, so an ability for this organism to cause invasive infection. And then there are also genes for biofilm formation, both in a patient but also in the environment. And I think this shows why this organism, A, can be so invasive, but also has the tools to colonize the environment. Genomically, it's most closely related to Candida lusitaniae and Candida hemolonii. This becomes a large problem for laboratories because these are two of the identifications that are most often misidentified as Candida auris, or Candida auris is misidentified as these two organisms. So it's divided up into geographic clades: South Asian, South African, South American, East Asian, and most U.S. isolates are mainly from the South Asian and South American clades. So I want to dive in specifically into lab identification. I think that we saw from the clinical case, there was a misidentification as Rhodotorula, but we knew it wasn't pink. There was also a misidentification as Candida hemolonii, and these are very, very tricky for a lab to be able to work out, and it's critically important for a lab to know whether or not they have Candida auris. And so this is an old table, so this is the review from 2017. We're going to talk about the updated identification schemes, but you can see whether it's API or Phoenix or Vitek or Microscan, even some of the MALDI-TOF, Vitek MS and MALDI Biotyper, previous to updates, were not able to reliably call Candida auris. In fact, for most of these systems, Candida auris wasn't even in the database. So ultimately, the identification could be an unidentified Candida, or it would misidentify it as a different Candida species. Now, the CDC actually has an excellent PDF around different identification schemes and their ability to identify Candida auris reliably. And I want to show off a couple of these for systems that have been used in the UCLA system. So starting with the Vitek 2 yeast. So the Vitek 2 yeast card, if you go down the right side of the pathway, the older versions identify Candida auris as either Candida hemolonii, Candida duobus hemolonii, or Candida species not identified. And if you don't have the current software version update of that, anytime one of those isolates comes off, Candida auris is actually a possibility. And at that point, it's going to require further workup by the laboratory to rule out Candida auris. Now, recently, Vitek came out with a software upgrade, and that software upgrade actually allowed it to identify Candida auris. So going down the left side of the pathway now, you can see that Candida auris can be confirmed. If Candida auris is confirmed on the Vitek with the software upgrade, you can see that the recommendations are to place the patient in transmission-based precautions, report to the CDC, and notify state and local health departments. Those systems can still misidentify Candida auris as hemolonii, duobus hemolonii, or Candida species not identified. So even in those cases, you do need to do further workup. And if Candida auris is possible, they suggest sending isolates to a reference laboratory, a state public health lab, a regional lab, or the CDC for further identification, and to place the patient in transmission-based precautions until that rule-out is confirmed. Now, at the UCLA Microbiology Laboratory, we use the bioMérieux Vitek MS MALDI-TOF for organism identification of yeast. Starting on the right side of the pathway, for older IVD libraries, so these are libraries before the most current IVD library update, you can see that Candida auris didn't exist in the system. So because of that, whether it's Candida hemolonii, Candida lusitaniae, or no identification, all of these required further workup. But once the new update came through, Candida auris can be reliably identified and confirmed. And with the Vitek MS, and this is true of the other MALDI-TOF systems as well, it's no longer going to misidentify that organism as hemolonii or lusitaniae. So a hemolonii or lusitaniae call on that system can now be reliable. So no matter what system that you use, and ultimately clinical microbiology laboratories use a wide variety of identification systems, the CDC has this great reference that I suggest you all download to be able to look at your own individual system and see whether or not that system can confirm Candida auris identification, or if not, what sort of follow-up testing is necessary. And you can see most of the identification systems are listed here. What's interesting is there are some phenotypic notes on the bottom. So let's say, for example, we look at the microscan system. The microscan system is not yet set up to identify Candida auris, but if it highlights Guermandiae, Parapsilosis, Lusitaniae, there's a note on the bottom that says these organisms generally make hyphae or pseudohyphae on cornmeal, going back to the experiment we saw with the case. If hyphae or pseudohyphae are not present, the isolate should raise suspicion as being Candida auris. So again, more of a workflow. However, some Candida auris isolates have formed hyphae or pseudohyphae. So again, all of this is requiring reference lab send out for identification for these particular systems. So it's interesting, if you have a system in-house that goes through one of the upgrades to be able to identify Candida auris, the laboratory needs to verify that identification. So it isn't enough that the system says it can do it. Now you as a laboratory need to test whether or not that's true. So we at UCLA recently upgraded our software to IVD Library version 3.2 for bioMérieux and our Vitek MS, and this includes Candida auris. So I'm going to walk labs through the process of how we then test for verification of this. We did an accuracy test. We contacted the CDC, and in the CDC's AR bank, or antimicrobial resistance bank of organisms, they actually can send you a set of organisms that you can test. So they sent us 11 different Candida auris, and then other isolates that are commonly misidentified, that Candida auris is commonly misidentified as. So 3 Candida duobus hemolonii, 2 Candida hemolonii, a couple of Saccharomyces cerevisiae, a Codame omeri, a Candida krusei, and a Candida lusitani. And when we did this testing, we found 100% of these isolates were identified correctly on the Vitek MS. What was also important for us to verify was different types of media. So as fungal cultures come into our laboratory, they can get plated out on different things, and that plating can lead to different levels of accuracy on your Vitek. So we tested species level identification on inhibitory mold agar. We tested it on SABDEX or SAB-high, so our brain, heart, and fusion agar. And then we did some precision testing, which was 3 isolates tested for 3 days in quadruplicates. All of this passed, giving us the sign-off confidence that if we have a Candida auris, we'll be able to recognize it to species in our laboratory. So I want to spend a couple of minutes talking about susceptibility testing and interpretation. So what we found in the clinical case is the Candida auris had to be identified correctly to species, but was also resistant to a lot of the known antifungals. So Candida auris has shown multi-drug resistance. It is probably a higher level of multi-drug resistance that we see in any other Candida species. Some isolates have demonstrated elevated minimum inhibitory concentrations to all available antifungal drug classes. And this includes azoles, amphotericin, and the echinocandins. So breaking out the isolates, over 90% of the isolates have a high MICs to fluconazole. So you can generally think of Candida auris as being resistant to fluconazole. Thus, fluconazole treatment without knowing the identification can lead to dangerous situations with Candida auris infections. 30% will be resistant to amphotericin. 40% of the isolates are resistant to 2 or more drug classes, and 10% are resistant to all antifungals. Only 5% are just resistant to echinocandins. So in that setup, I think that treatment changes before you identify whether or not the full species identification of the yeast has been moving from something like an azole to, in areas of high Candida auris, something like an echinocandin, where there's a lower chance of resistance. Interestingly, antifungal susceptibility testing for Candida auris is not standardized. CLSI has not put out full Candida auris breakpoints. And so when looking at a table here, and this comes from Journal of Clinical Microbiology paper in 2018, Candida auris an emerging fungal pathogen, we can look at some of the MIC ranges and then have a list of potential resistant breakpoints. So for fluconazole, the idea of a potential resistant breakpoint is greater than 32. The MIC range of all organisms for Candida auris tested was 0.12 to greater than 64. But an MIC 50, and this is an MIC that allows for killing of 50% or greater of the organism, was all the way up at greater than 64. So this shows that this organism is almost routinely highly resistant to fluconazole. We see some variation in some of the other azoles, the echinocandins, including anidulafungin and caspofungin, and then also amphotericin. And so the MIC ranges are broad, but we can definitely see resistance to those antifungals as well. So all of this paints a fairly disturbing picture, right? We have an organism that can cause invasive disease, contaminate the environment, is very difficult for the laboratory to quickly identify as Candida auris, and susceptibility testing is not routine and doesn't have good breakpoints. So what can we then do as a microbiology laboratory to help stop this outbreak? So number one, you want to be able to ensure that your lab uses a yeast identification system that can accurately identify Candida auris from a clinical sample, or knows where its own identification system is weak, so it knows when to send an isolate out. The key part of all of this is really a race against time, right? Once a Candida organism is isolated and identified, the longer it takes to identify it to species, the more likely that Candida auris can be passed around the hospital, the more likely that invasive infections can occur. And as you saw before, sometimes it only takes 48 hours to be able to have a patient get colonized and have an invasive infection from a Candida auris in the environment. So we need to work as laboratories to be able to identify these organisms accurately to species as quickly as possible. In doing that, you want to be sure you perform a laboratory verification to make sure the system is both accurate and precise. And you want to make sure yeast susceptibility testing is available for clinically relevant isolates. This can either be in-house, which is more unusual. We do in-house testing at UCLA for Candida, or as a send-out to support clinical treatment. This really helps in the case of identification as well. So if you have a Candida that couldn't be identified as species, but you're able to do some susceptibility testing, potentially you can guide therapy before that full species identification comes through. So another thing a laboratory can do is called passive surveillance. And this is an idea of how do we, for any yeast within the clinical microbiology laboratory, try and identify Candida auris. So I would say many or most clinical microbiology laboratories only identify yeast to species from sterile sites, blood, tissue, CSF. But in non-sterile sites, genital, urine, throat, they're usually only identified as yeast on the lab report, and this is enough to help with clinical treatment. The challenge here is that this allows for a potential missing of a Candida auris infection. And so state and county public health laboratories are suggesting that clinical microbiology laboratories start attempting to identify these organisms to the species level, especially for inpatient. What we found in the outbreak in Orange County is that it was this level of passive surveillance and the identification of a yeast species as Candida auris in the urine, which led to understanding that there was an outbreak going on. So we recognize, though, that this is a significant impact on workflow for a potential mycology bench. So at UCLA, we're identifying the species all yeast that are ordered as fungal culture. But if a yeast grows up on a bacterial culture, which can routinely happen, especially in things like urine or respiratory, we would either identify that as yeast, or if it was low enough quantity, it's just part of the normal flora. In moving forward with passive surveillance, it's not that we're going to identify that yeast species if it's not Candida auris for clinical care. But what we want to be able to do is test all of them to rule out Candida auris. So at UCLA, there were about 2,600 cultures, positive cultures over the last 6 months that fit into that particular category. And of that, 200 of them had a yeast reported. So this is really a movement of 400 additional yeast identifications onto a mycology bench. We are a medium-sized academic center and clinical microbiology laboratory. We recognize that these sorts of suggestions may be very, very difficult to follow up on in the large reference centers that have many more cultures, but we do agree that this is one way to monitor your patient population to see whether or not Candida auris is in your hospital. Another thing that the lab can participate in is something called active surveillance. And active surveillance is really this notion of screening patients for colonization on admission to a hospital. What high-risk patients can you look at coming in to know whether or not they have Candida auris and put in the appropriate infection prevention measures to be able to stop that from being moved around your hospital? So there are CDC guidelines to this on who should be screened for Candida auris. So CDC guidelines dictate close healthcare contacts of patients with newly identified Candida auris infection or colonization, including roommates at nursing homes. Additionally, patients who have had an overnight stay in healthcare facilities outside of the United States in the previous year where there have been documented cases of Candida auris, and as you can see by the list below, that is a lot of different countries. Now, this ends up being challenging for a hospital, being able to recognize on intake whether or not one of these two things have occurred. But if they can recognize them, surveillance is suggested. So how does active surveillance occur? How do you figure out whether or not someone is colonized with Candida auris? So the suggestion from the CDC is to screen with a single composite swab of the bilateral axilla and groin, and this is called a Zorro swab or a Z swab, and you can see it goes across the armpits and over the groin. These are the most common and consistent sites of colonization, and these have been used, this sort of screening has been used in the outbreaks in New York, in the outbreaks in Chicago, to be able to recognize colonized patients. The question then is, how then do you figure out whether there's any Candida auris in that specimen that was taken? So right now, screening by culture-based methods are available as a send-out to the AR lab network. So the AR lab network is a series of labs in the LRN. So on the West Coast, our laboratory is the Washington State Public Health Lab. And in this setting, if there's screening of a high-risk patient, you can send that sample to Washington State. You submit the eSwabs on ice within 96 hours of collection, and they will culture to be able to figure out whether there's any Candida auris in there. This is a fantastic service and is available to any clinical laboratories within that region. Part of the challenge of this service, though, is that it's a 1 to 3 week turnaround time. Culture methods take a long time, and from an infection prevention standpoint, if you're going to have a patient in isolation for 2 to 3 weeks before you figure out whether they have Candida auris, that's okay for one patient, but that may extend even beyond their stay in the hospital. And if you have 40 patients that now need to be screened, you may not be able to keep that many patients within isolation or cohort them together because you don't know which one has Candida auris and which one doesn't have Candida auris. So this represents a challenge for both hospitals and clinical laboratories. You can screen by culture for Candida auris in-house, and so as a microbiology laboratory, not send out, but current culture surveillance recommendations include inoculation onto several selective agar plates and enrichment broth to be able to increase sensitivity. Once those are done, you still need to hold the culture for up to 2 weeks to actually rule out a negative. So unfortunately, culture in and of itself, it's an extended workup, it's difficult for laboratories to perform, and we think that the length of culture time is suboptimal for surveillance and infection control efforts. So we feel there's an urgent need for a clinical PCR test for surveillance of Candida auris that can be rapidly implemented in many clinical microbiology laboratories, and we've been working on developing that here at UCLA. So we've recently developed and are using a Candida auris surveillance PCR that offers a rapid turnaround time, so the test can provide in 4 hours test results on 12 samples, and the ability to include up to 94 samples in a run, which is advantageous in a healthcare setting when high volume surveillance is needed. I want to go into some of the details on the development of this particular test. And so in targeting the PCR assay itself targets a highly conserved region of the ITS2 gene in Candida auris. And this PCR assay the primer targets, were tested for cross-reactivity against a number of different organisms. So almost all the Candida, and this ends up being super important because Candida is an organism we're all colonized with, right? It's part of our normal microbial flora, and you don't want a false positive Candida auris just because Candida albicans is there. Other yeast species as well, other molds, other bacteria, even other viruses, just to look for cross-reactivity, and there was no cross-reactivity that was seen. Then in that setting, we needed to put together a PCR protocol. And so we put together a protocol not only for colony suspension from culture, but also from the eSwab that will be taken directly from the patient. Briefly, just to go over this, we resuspend a pelleted eSwab in 0.5 mils of lysis buffer, transfer that, do both bead beating, vortexing, and heating. And all these steps actually are critically important in being able to get a sensitive PCR assay. The Candida cell wall is a tough component. And if you don't break that cell wall open and reveal all of the nucleic acid that's there, it can greatly negatively affect the sensitivity of your assay. So we go through a substantial pre-extraction step, and then we extract DNA on a QIAGEN platform, the EZ1 DNA platform, and then we have some positive controls and negative controls, of course. So Candida auris strain is a positive control. Negative control is eSwab matrix alone. Moving on, we worked with DiaSorin to be able to put this test working on the Liaison MDX platform. It's a platform that we at UCLA have numerous laboratory-developed tests. And so in that setting, the assay definitions are there, the cycling parameters are there, and we built out the assay to be able to give us a sensitive and specific PCR. So really building out the assay is the first component. The next component is how you have a clinical validation. So understanding whether or not this is going to work clinically. I wanted to walk the audience through our clinical validation. So we took negative e-swab media, and we had a healthy male adult volunteer living in Los Angeles, where at that time there were no Candida auris cases reported, who agreed to have his armpits and groin swabbed numerous times to be able to fill out a negative e-swab media. So now we have media that we can spike in Candida auris for identification. Then we spiked in Candida auris at varying dilutions, ranging all the way from 10 colony-forming units, very, very sensitive, all the way up to 10 to the 7th. And then in our negative specimen, we were able to spike in other yeasts that we knew potentially cross-react, Duobus hemolonii, Lusitaniae, at a high concentration, and then did a seed in recovery. So the results are shown in the table. Of 141 expected positive Candida auris by seeded recovery, we had 141 that were positive by PCR. Of the 84 that were negative, containing other organisms, all 84 negative by PCR, giving 100% sensitivity and specificity and concordance across the board. Another interesting question is, what's the limit of detection for this? So how low can we potentially go and get usable signal? So spiked samples with serial tenfold dilution of Candida auris were tested to determine the LOD, defined as the minimum concentration with a detection of at least 95%. So the LOD was determined to be 1 times 10 to the 2, which brings us into the range of other LODs for PCRs and is very acceptable from a sensitivity perspective. You can see on the table the mean CT values going all the way from 17 at 1 times 10 to the 7th, all the way down to 35 at 1 times 10 to the 2, giving us a 100% detection rate above 10 to the 2 and above. We also needed to do some precision testing. That precision testing went relatively quickly, so we tested at the LOD and then also at a high concentration, and you can see there was 100% detection across the board. And then we wanted to do some stability. How long could it last in a specimen before we tested? And the stability was constant across the board. We actually even saw some organism growth at room temperature. And so that led us to the conclusion that we have successfully developed and validated a clinical PCR test. The laboratory developed test showed excellent performance, and we hope that since the reagents and instruments described in this validation are commercially available as ASRs for LDT development and widely used, many other clinical microbiology laboratories could take this on, use our validation protocol, and then bring in their own in-house PCR. And so to wrap things up, in conclusion, I take this from a news article. 4 things to know about Candida auris. It's a drug-resistant fungus. It's difficult to diagnose. It shows outbreaks in hospitals and nursing homes, and it's preying on elderly patients that are immune compromised. Clinical microbiology laboratories are critical in the fight against Candida auris, and labs need to be able to use a fungal identification system that can accurately identify Candida auris to species. This is important for invasive infection, but also colonization. And labs can practice both passive and active surveillance to assist in hospital infection prevention and recognizing colonized patients. A lot of work went into this. I want to thank the UCLA Clinical Microbiology Laboratory. I want to thank the UCLA Infection Prevention Team. I want to specifically thank Sean Yang, Evan Hilt, Travis Price, Brian Bolin, and Ruel Mearsall, who were specifically involved in the clinical validation and the forming of the PCR test. And I also want to thank the group at DSOR Molecular that assisted. And I, at this time, am open to any questions.
Thank you, Dr. Garner, for that presentation. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your presentation window, labeled Ask a Question, and then click on the Send button. Dr. Garner will answer as many questions as time permits. So let's take a look at our audience questions coming in. Dr. Garner, since we know that Candida auris can survive on surfaces, do you have any suggestions for sampling surfaces? And what steps would you suggest for those facilities that have colonized patients with active infections as it relates to ensuring that they have ruled out surface contamination?
This is a great question. And so what I didn't mention in the validation of the PCR, because it was beyond the clinical work, is we actually had validated that PCR for environmental testing as well. And so what we see in our system is that you can swab areas of a hospital room, be it the bed rail, the vitals, the floor around the patient, put that into the eSwab media, the same eSwab media that we're using for active surveillance, and PCR positivity can be obtained from that. So PCR represents a really good way to real-time look at environmental contamination in a room. Culture of that is very difficult, right? It's the same problem we saw before. So if you're going to attempt culture in-house, or you're going to send out to one of the state reference labs to be able to do this, it's still going to be a 2 or 3 week turnaround time for surveillance. So I think ultimately PCR is the best way to go for environmental surveillance if a laboratory is able to do it, or if a local public health laboratory is able to bring on a PCR test.
Thank you. And since Candida auris can be shed by colonized patients and healthcare workers, which allows the opportunity to contaminate hospital environmental surfaces, will infection control practitioners target both colonized patients and contaminated surfaces as part of their infection control measures?
The answer to that is absolutely yes. And so if a patient's going to be positive, the patient themselves is going to be tested. The room around that patient is going to be tested both before cleaning and after cleaning to be able to make sure that not only is the infection cleared from the patient, but also whether that patient is continued to be colonized and whether or not the room itself is contaminated.
Thank you. And I want to thank our audience members for these great questions coming in. Dr. Garner, our next question is, when did you complete validation of your active surveillance PCR, and have you detected a positive case?
So that's another great question. So we completed validation about a month ago and went live with our test. And we actually have had transfer of a patient who was known Candida auris positive to our facility from the Orange County outbreak. And we were able to then validate not only our surveillance swab on a patient, but also environmental testing from that particular patient. So we have had a colonized patient at our facility that was being watched by infection prevention, a known colonized patient.
Thank you. And once a positive is detected by the PCR, what are the next steps at your institution, and how is the patient then treated?
That's a great question. So our patient wasn't from an infection, and so it wasn't from a clinical specimen. So in that setting, because the patient was colonized, it's really, one, getting that patient into an isolated room. We're fortunate enough at UCLA that all of our rooms are single occupancy rooms, but if they weren't, then that patient would have been needed to move from a room where they were cohorted to an isolated room. I think that's the first step. And then the second step is really after clinical care. So once the patient is treated for whatever they're there for, once they leave, it's a process of surveilling the room to see whether or not it's contaminated, cleaning the room, and then testing the room again to make sure that that organism isn't there for the next patient to come into the room.
Thank you for that. And we have time for a couple more questions. Does the diazorin primer pair detect all of the clades of Candida auris?
Yeah, that's a great question, Sue. So the diazorin, it does. It recognizes all of the clades, but it doesn't differentiate them. So they would all show up equally Candida auris positive, but for differentiation, it really requires whole genome sequencing, and that's something that is typically beyond the scope of a clinical laboratory, but often can be done in a public health laboratory for epidemiology and understanding where the outbreak came from.
Thank you. And Dr. Garner, we have time for one more question. Our audience member wants to know, can the PCR test be used for both passive and active surveillance?
Yeah, I think that's a great question. I think that in general, the answer is yes. So what you could do is grow your yeast, let's say the yeast that you identify on a urine culture that you're just going to call yeast, and you could run PCR on that. The challenge in using it that way, though, is that's an expensive way to use the test. So ultimately, because you already have a colony, there are other cheaper identification systems, especially MALDI-TOF, that can reliably identify that organism to species, and you can just keep PCR for the non-culture method, so for surveillance, active surveillance, and then also surveillance of the environment. I think that it would be more cost-effective to use it that way, but ultimately the PCR would also work on isolated colonies of organisms.
Thank you. And Dr. Garner, do you have any closing remarks that you'd like to provide for the audience today?
I just want to empower microbiology laboratories and laboratorians to be involved. We are a critical part of not only lab medicine, but also stemming outbreaks, helping to diagnose infectious disease, and laboratories bringing themselves to the place where they can confidently identify Candida auris and assist their infection prevention really is going to be one of the major things that stops this outbreak from proceeding across the entirety of the United States.
Wow, thank you so much for your time today, Dr. Garner, and thank you for this important research. I'd also like to thank audience members for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of your registration. And I'd like to thank Labroots and our sponsor, DiaSorin Molecular, for underwriting today's educational webcast. This webcast can be viewed on demand, and Labroots will alert you when it's available for replay. We encourage you to share this email with your colleagues who may have missed today's event. Until next time, bye-bye.